It's a little bit overwhelming because most of my mentors over the last decade are in this room right now. Jan, Marlene, Pat, everyone who's kind of guided, not just me personally, but PTC through our trajectory in rare diseases. And we get credit for being very clever and reaching out to patients early on. The truth is we had to because we didn't know what to do. We had a mechanism of a drug, which I'll show you a little bit about. We didn't have an indication selected. So we had to go and talk to the patient groups from the beginning to even decide which path to follow. And little did we know that this would be such a productive way to engage, so enriching to us personally as people, but also for the company in really building something that I think is consistent with the interest of who will always be our most important stakeholders. So in putting together this talk, we put together our slides, and then I realized that the titles was the challenges and the opportunities. And I really feel like our slides fall short on the opportunity side. So I'm glad that Sarepta comes next. I hope that they'll nail that for us, because they're certainly making that come true and getting a lot of attention for Duchenne muscular dystrophy. But having worked in DMD in the last decade, I think I'm better equipped, honestly, to focus on some of the challenges along the way and hope that this experience can translate into something that's useful across other areas. So some of the opportunities, just in a general sense, I think we can agree that for us who care deeply about science is the idea that we can do things that are intriguing to us. And we see this at PTC with our work in ataluren, which is focused on nonsense mutations, but we also see in our earlier work in spinal muscular atrophy or in other genetic disorders that we're targeting. The idea that you can impact patients' lives so directly and you actually know who they are, it's very, very meaningful. But also, of course, there are some incentives or market exclusivity and the premium pricing and other elements that have proven over time to show this to be a sustainable and very attractive business. Now to the challenges. So I think when people think about small patient populations as being desirable, I always think about the idea of flying from New York to Boston. It sounds like it should be faster than flying to Europe, but it sometimes isn't. You know, you get there and there's delays here, there's just delays in the air and the delays landing, and it's 7 hours later and you're just a couple of hours away. And I think rare diseases have some of those components where something looks straightforward and you really think there should be no reason why this should be so complicated. And I think we've had a lot of examples of things that turn out to be more complicated than we expected. Finding your patients, it's one of them. If you have a small population, you now have to locate them. If you happen to be similar to PTC, where you need patients with one specific type of mutation, then you have to look for a small population within a small population, and that itself is a challenge. If you're looking at an endpoint, that eliminates maybe some of those patients within that population. Now it's narrowing even further. So we really had to assess for our phase 2B and now for our upcoming or ongoing phase 3, every single site we came across around the world in order to identify enough patients. The lack of therapeutic options can create this obvious desperation from the patient groups, and managing those expectations are very, very difficult for everyone involved. It's a great amount of motivation, but it's certainly something that you struggle with every day. And the fact that I think in this room, it's recognized that a clinical trial was really to determine whether a drug has a benefit, not necessarily as a means to a treatment, but for those patients who are desperate for treatment, that clinical trial is viewed as access to a potential therapy, and that expectation is very, very heavy, even to just participate. So moving on into some of the challenges, once you have your actual population, and we worked very closely with the patient groups, not just in Duchenne muscular dystrophy, but also in cystic fibrosis that's had a registry from the 60s. I remember being on the phone with the Cystic Fibrosis Foundation and hearing the clicks when my colleague was looking up, seeing if they had enough patients with Duchenne muscular dystrophy to do a US-only trial. And he said, oh, you definitely need a global trial. We ended up doing a global trial in CF. And in DMD. But then now you have to figure out what is the path to develop that drug in a way that will make sense and show a real clinical benefit and be acceptable to regulators. When we started working Duchenne muscular dystrophy, I had this fabulous associate, Carrie, in my team, and I asked Carrie to go pull up everyone who got a drug approved in DMD in the last 10 years, so I could just look up the endpoints and the investigators. And she came back a couple of hours later, very pale, and said, I couldn't find any. And then I thought, wow, you have to do everything yourself. And sat there at night and drank some coffee and went through it. And much to my shock, this is 12 years ago, there hadn't been anything even really pursuing endpoints. And we went to explore these endpoints with the community, and we found this to be very controversial because everyone had a favorite endpoint and no one seemed to agree. So getting that started was definitely very, very challenging. And then, of course, getting the blessing from both FDA and EMA in terms of, can this actually be the basis for approval? It's not a trivial task. Then taking that design and translating into a trial that's executable from the point of view of something that you can see maybe in your proof of concept studies in a number of sites, and then taking that into something that's global, in our case, 37 sites in 11 countries, definitely poses some logistical challenges that we were unprepared for, quite frankly. Looking more specifically into the development of ataluren in Duchenne muscular dystrophy, I think hopefully everyone in this room now has heard of Duchenne muscular dystrophy. We've been working on it since a time where everyone confused it with multiple sclerosis, so it's great that it's more recognized. It is still an area of great medical need, and the encouraging part is that there are a number of things in development, and we hope that they will turn out to be viable therapies. But what is discouraging is that even with everything that's in the pipeline today, we probably don't cover fully all the patients in DMD in an immediate 5 to 7 year scenario, so more development is needed. Ataluren, that I'll talk to you about today, focuses only on patients that have the disease due to a nonsense mutation. So in Duchenne muscular dystrophy, that's approximately 13% of the patients, so that is pretty small. The disease is relentless, it's well conserved across populations. You see symptoms in 2 to 4 years of age. Progression goes to becoming non-ambulatory or dependent on a wheelchair in the early teens, and most patients don't survive into their 30s. So it's definitely a very severe disease. How our drug works, and this is the reason why we had a mechanism before we had an indication. Dr. Stuart Peltz, our CEO and the founder of the company, had done his work focusing on the mechanisms. I guess if I point, it would be difficult to see the mechanisms of how a nonsense mutation, which you can see here by the yellow stop sign, is different than a normal termination codon, which is the red stop sign. And the theory was, in his mind, that if you could differentiate between one and the other, you could get a drug that would read through the inappropriate or incorrect termination codon and go on to make a full-length protein, stopping at the appropriate termination codon. And that's exactly how ataluren works, just taking about 20 years of his life. But the molecule comes in and basically allows the protein to be made by allowing you to read through that first inappropriate termination codon, make a full-length protein. A lot has to do with the fact that the specificity of this mechanism has to do with the fact that we're drawing here for you, this globally glob, as I like to call it. He doesn't like that term. It's the ribosome making the protein, and it has to do with the fact that when you terminate that process, you're actually very close to the reinitiation. It's very, very circular, and it's very well conserved, while the premature termination codon is kind of sticking out there, not a very difficult thing to read through. That's what allows us to bypass that and still stop at the normal termination codon. But this is a mutation that can cause disease on, as far as we could tell, any genetic disorder on a percentage of the patient. So as far as we could tell, you take any genetic disorder and you look at a number of the patients, about 15% on average have the disease due to a nonsense mutation. So like I mentioned, 13% in DMD, it's about 10% in cystic fibrosis, and there's a large number of other disorders. Here's something to be excited about. There are a number of different things, as I mentioned, being developed in DMD, but PTC's approach, ataluren, doesn't overlap with the approaches of Prosenza, GSK, or Sarepta, where our patients that have a nonsense mutation would not benefit from their approach, and we don't believe their patients would benefit from our approach, because this patients that have a nonsense mutation are separated from the patients that would benefit from exon skipping. If there is further development and exon skipping is applied over and over again, you can certainly consider applying it to patients with a nonsense mutation, but hopefully they have a long enough list that they will be focused on other things. This is a slide that was made by some folks on our commercial team, and I think it really summarizes beautifully the challenges that we have along the way. I won't read through it, but it just comes together from the fact that the disease is rare, locating all the patients, the determination of the clinical path, and some of the challenges that we've had in our clinical trials as it relates to the statistical model that we used, and realizing that we should have used something different. Of course, hindsight is always 20/20. And having to develop that path while trying with the same population where we're still doing dose finding and hoping that it could serve as the basis for approval. Certainly a large challenge. The 6-minute walk was the endpoint that we decided at PTC that would be the most appropriate to be used in Duchenne muscular dystrophy when we started to look at the list of endpoints that were being proposed by our investigators. As you know from Duchenne muscular dystrophy, I mentioned to you that the disease is progressive, and there is a period of time where you start seeing this progress in, or decrease, I should say, in ambulation. The goal is, of course, to treat patients across the entire spectrum of the disease and certainly delay it or prevent it from developing as much as possible. But we have to find an endpoint that we could measure in the clinic in a way that was consistent, and again, that could be done in a number of sites. And this is where the 6-minute walk proved to be very useful. We developed a training video sponsored by Parent Project Muscular Dystrophy. We used this video in all the visits in all our clinical sites. It was very detailed, and it really allowed the investigators to perform the test in a very consistent manner. And we did a study with Dr. Craig McDonald, who's an expert in the use of 6-minute walk in Duchenne muscular dystrophy, to test the feasibility of this endpoint. One of the concerns that the physicians had very early on were 6 minutes for a young child can be a long time. Would they really stay focused? Some of these patients have some issues with autism spectrum disorders. They may be distracted. Some have contractures on the legs and maybe not be able to perform the study. So there were a lot of concerns as to whether this could be done. I think the study that we did showed that the endpoint could be used, and from there we went on to build our phase 2b study, which had still 2 different doses of ataluren, well, called the low dose and the high dose, given 3 times a day. It's an oral drug. It's a powder that is mixed with water or juice or applesauce and swallowed 3 times a day. We stick to meal times for convenience. We stratified the study based on age, the use of corticosteroids, which at the time was not as common as it is today. It certainly evolved very rapidly, which is wonderful because we're thinking the standard of care is getting applied more broadly. And we looked at their baseline levels on the 6-minute walk based on what we knew at the time, which, as you saw from the previous slide, was very little, right? The actual 6-minute walk performance in Duchenne muscular dystrophy was very limited. So there were 174 patients, as I mentioned before, and here are the results. So what we see on the green is the low dose, where we see a difference of 31 plus meters, and that exceeds our target of 30 meters, and that's very encouraging. It was very surprising to see on the high dose in blue that we did not see a similar effect. In fact, while it actually looks like placebo is better than the high dose, when you look at the numbers on the right, it's actually exactly the same. So the high dose had no effect at all, and it really puzzled us. It took us a long amount of time to figure this out, and we think we understand this now as being related to the mechanism of the drug, and it has to do with the way that the binding of the ataluren molecule works on the ribosome, And the idea is that if you have too much ataluren, when you're binding to the low affinity binding site and the high affinity binding site, you basically block the effect of the drug. And it's hard to accept an instance where more is not more, but it turns out that this is reproduced in other areas as aminoglycosides that have similar mechanisms. So after about 2 years of thinking about it and doing some experiments, I think we validated that what we call the low dose is really the efficacious dose here, and it is the dose that will be moving forward. The other thing to point out is that the consistent of the exposure that we had in the low dose was consistent with what we've seen in animal models of what we were hoping to reach in the human exposure. We know that 30 meters in general has been the basis for approval in other therapeutic areas, so we had hoped that this would still be the basis for approval for ataluren. That is still what we're targeting. We think this is something that has been accepted by both FDA and EMA. I will say that we constantly still get questions about what do 30 meters really mean for a patient with Duchenne muscular dystrophy, and it's interesting because from the patient side, what we hear is forget 30 meters. It's the 5 meters between here and the restroom, the 10 meters between here and the next classroom. That's what they're looking for, but it still seems like we need to explain to regulators how patients would perceive this benefit and perceive the idea that their disease would not progress as aggressively, but there's definitely more work to be done on that. So what did we learn about the natural history of DMD? And I think this is really the greatest outcome, not just for ataluren, which is progressing well, but for the entire community. What we learned is that the parents were right. What a shock. And that you have in DMD this period of time that is often referred to as the honeymoon period, where it seems that the child is not decreasing in function. The walking ability doesn't seem to be declining, and that's because the cognition and the maturational development of the child are basically overtaking the progress of the disease, and you don't see a decline. Then you have a period of stabilization where things kind of come into more of a status quo, and then a rapid decline of the walking ability. And that is probably a good area to start measuring that change somewhere along the lines, but certainly not when you're looking at the maturational improvement still taking over from the disease. What we see here is that when we took our population, in the trial we had patients from 5 years and older, but when we actually just took a population and looked at the patients over 7 that were on steroids, and the steroids didn't improve the amount of walk, but it did make them more stable. When you looked at the population alone, we had a much stronger benefit, almost 50 meters. So it does tell you that it's something that we could measure in that population, more so than when we included the 5-year-olds. That's definitely a big change. Another thing that is striking is the change that you see when you're looking at patients who are higher than 350 meters on their baseline. So basically, if the patients coming into the trial, they had to be approximately 7 years of age, or they were 5 in the phase 2B. But if you thought about the patients who were about 7, and you look at the patients that were below or higher 350 meters in their entry criteria, we saw some elements of the natural history that we didn't know. Strikingly here is that the patients that are walking over 350 meters really don't have much of a change in the period of time of 48 weeks, while the patients that were below actually have a very rapid decline. So we think we found kind of the sweet spot where we can measure this, where we can use this endpoint in the most meaningful way. Looking at greater than 7 and greater than 350 meters. So patient, it's 7 or older and at a baseline walking at or more than 350 meters, what we saw at 48 weeks is they basically didn't change at all. So that's obviously not a good patient for the purpose of this study. And it does tell us something about a natural history that we can apply across other interventions as well. And the change when we see under 7 meters, but where you're still greater than 350 meters, it's almost as if they're improving on their own, which we know for a fact that they're not really, but maybe a little bit. So the lessons learned here for us, certainly the variability on the progression was, I think, larger than we had expected. The effect seen on the high dose was not something that we had expected. It was definitely a lesson learned, and probably goes to the fact that we still called it a phase 2B because we did have phase 2 doses in it. And then working through the community to have the acceptance and have the 6-minute walk be embraced, which was very controversial at first, but I think now has become really the standard across trials in Duchenne muscular dystrophy, which is rewarding. The next steps for ataluren specifically are phase 3 in Duchenne muscular dystrophy as well, where we now have refined this criteria to take into account what we learned. So specifically patients who are over 7 years of age and they are below 16 years of age, they have to meet all sort of detailed criteria in terms of the signs of DMD. They're shown by age 6, and that's detailed on clinicaltrials.gov, but they are all on steroids now, and that is a stabilizing factor, but it also reflects that more and more patients are on steroids now. The standard of care has progressed, and that's a good thing. And then really narrowing in on the 6-minute walk, where the patient should be at an 80% predicted. We can now calculate, based on your age and height, what your 6-minute walk results should be a baseline to make sure that you are in an area where we can measure a change within a 48-week treatment period. So we're excited about the study. We think this will go on to reproduce the results that we saw in a similar population in the phase 2B. And this completes my presentation. I wanted just to point out that when we think about opportunities in DMD, we have a saying with our scientists that there was a little boy with DMD that had heard that the gene had been identified. So he drew up a calendar and drew a picture of himself playing baseball and because he heard that about a year later there should be a cure. So when you think about anything that you're doing in rare disorders, think about your audience, not just being, of course, investors in the community and all those that we want to keep excited, but the little boy who is writing that down and pasting on his wall. He's not around anymore, but his mom is still fighting for him. So thank you.